Welcome to Beyond Lab Walls, a podcast from the Salk Institute. Join hosts Isabella Davis and Nicole Milnarek on a journey behind the scenes of the renowned Research Institute in San Diego, California. We're taking you inside the lab to hear the latest discoveries in cutting-edge neuroscience, plant biology, cancer, aging, and more. Explore the fascinating world of science while listening to the stories of the brilliant minds behind it. Here at Salk, we're unlocking the secrets of life itself and sharing them beyond lab walls.
Today we welcome Pam Maher, who is a trained biochemist and cell biologist that spends her time at the Salk Institute studying the intersection between aging and neurodegenerative disease. Welcome to the podcast, Pam. We can get started at the very beginning. Where were you born?
Well, I was actually born in New York City, and I lived there for about 6 months. And then my parents moved to the suburbs in Connecticut, in Fairfield County.
And did you stay in Connecticut for college?
I went to McGill University in Montreal because I wanted something really different. And I also didn't want to be in a school that was, at that time, there were still all-female schools, and it was not my idea of a good time. And also, I wanted a city rather than a small town because I had grown up in a small town, and I didn't like the environment of small towns where everybody knew everything about you.
Moving to Canada, you must have liked the snow?
Well, I had grown up with snow, so the snow didn't particularly bother me. I was used to that, cold winters, although Montreal was definitely colder than Connecticut.
Do you remember being interested in science or plants or anything when you were a kid, or did that start later on?
Actually, it's so I was really interested in science, and then I had a couple of science teachers in middle school and high school that I didn't really like. And so I got kind of turned off from science. And when I went to college, I was thinking of majoring in political science. The woman in the room next to me was going to major in science, and she would come back and tell me about her classes every day. And she was so excited about learning all this new stuff. And I was not very excited about my classes. So I thought, well, maybe I'll just try some of her classes because they sound a lot better than mine. So I just started going to her genetics class and then decided I'd transfer into biology.
Was it difficult to switch majors?
That presented some hurdles because the genetics class was actually a second-year class, and she had taken advanced placement biology, so she was able to skip that, but my school didn't offer it, my high school. And so I had to, at the same time I was taking genetics, I had to go backwards and take first-year biology. And then I decided, so in the biology area at McGill, there were all sorts of options. You could major in like marine biology or cell biology, but biochemistry was sort of considered the most rigorous. And I figured if I'm going to do this, I might as well challenge myself.
So at what point did you decide you wanted to pursue research after college?
Well, the people around me, they were all pretty serious students. So I was surrounded by people who were planning on continuing, a number of them in either medical field or in basic sciences. So I think that kind of became the direction I figured I would take as well.
Did you consider medical school?
I did, but I'm really not, I don't have a lot of empathy. And so I just didn't think I was going to be very good. I would not be very good with patients. I wanted something where there was more discovery, I think. Thought staying in biochemistry, cell biology offered more opportunities for sort of exploring and discovering.
And then did you stay in Canada for your PhD?
Yes, I did. Yeah.
And what were you looking at during your PhD?
I ended up in the lab of a guy who really mostly interested in the eye, but he was just setting up his lab at the time. So he was open to exploring new areas. And I was more interested in nerve cells. One of the things he had developed was a technique for labeling cell surface receptors and being able to visualize them. So you would tag little spheres with antibodies or other molecules and look at their distribution on the cell surface and how that was changed with different types of treatments and stresses and things. So that was one of the areas that I pursued, was to look more at how different types of stresses change the distribution of these different receptors on nerve cells. And that led me to San Diego, because one of the people at the time who was at the forefront of looking at the interaction between cell surface receptors and intracellular network of actin and microtubules was John Singer, who was at UCSD.
And how was the experience coming from New England and Canada and going to San Diego?
So I had been in Vancouver, which for Canada is kind of the San Diego of Canada, I guess you would say. It's more temperate, and it rarely snows there. So I had already kind of moved from really cold winters and a lot of snow to Vancouver, where the winters weren't really that cold, but they were pretty dreary. So it was actually nice to come here and have more sun. Yeah. And obviously I liked it because I'm still here long time later.
Yeah, I was going to say, you've stuck around. So San Diego has grown on you.
Yes.
So what was it like at UCSD and other places that you were in San Diego that weren't Salk?
So UCSD Singer had a pretty big lab, I think. It was great because they were from all over the place and a variety of different backgrounds, and in general, pretty sociable, really welcoming. Somebody seemed to always be organizing something. And so it was a really good experience. And then the lab kind of over time shrank a bit. And then I ended up going to a small place that was at the time affiliated with Scripps Memorial Hospital, called the Whittier Institute, which I don't think it exists anymore. But the reason I went there was that Roger Guillermin, who had moved there, and we were working specifically on fibroblast growth factor receptors, which had some of that early work had come out of Guillermin's lab. Then the Whittier, I guess you would say, kind of fell apart. The person who had started it retired, so we were able to negotiate moving to Scripps Research Institute, and so we did. And then I was there for about 8 years, I guess.
And how was it there at Scripps?
The person who had agreed to take our group in, was in the cell biology department at Scripps. He died. And the new departmental chair wanted to take the department in a different direction. And she basically told me, I'm not interested in your work, so you should leave.
Brutal.
So at the time, my husband was working, Dave Schubert was working at Salk. And he said, why don't you come here? So I did, initially as a senior staff scientist. So it was much better to come here and work in Dave's lab with Dave and pursue my own interests, which were more closely aligned with Dave's. And I think it gave me a lot more opportunities to expand in the areas that I was more interested in.
So what exactly was it that you were studying in those years as you transitioned into Salk?
I ended up focusing more on fibroblast growth factor receptors and their signaling mechanisms, and particularly one of the fibroblast growth factors called fibroblast growth factor 2, or basic FGF.
Fibroblast growth factor receptors are little proteins that live on the surfaces of cells, which are crucial in regulating the development of cells, then maintaining cell function once they're mature. Fibroblast growth factors are the key to the fibroblast growth factor receptor lock, and the interaction of the two is frequently implicated in cancers.
That led me to begin to start asking questions, which there was some evidence that this could be protective in nerve cells. At the same time, we were doing some studies, Dave's lab, and then I was interested in this too, of developing models of nerve cell death. So one of the obvious things to start looking at was fibroblast growth factors. It turned out they didn't help at all in protecting the cells. And then Dave had a postdoc in his lab who was really interested in natural products. He started looking at the protective effects of natural products. And I thought this was really interesting, and I was really impressed by some of the data he was generating. And so that was the beginning of our work with looking at natural products as neuroprotective compounds.
And when you came to Salk, you were in your husband's lab? How did you meet?
We were both doing triathlons at the time, and we were in a master's swimming program at UCSD. And we were both in the slow lane because neither of us were very good swimmers. So Dave and I were in the slow lane. And so we would talk we were catching our breath between sets, and it turned out he was at Salk and did some interesting research. And it turned out from talking, obviously we had both had an interest in triathlons, and we also had a common interest in science.
What was it like collaborating scientifically at Salk?
His lab and now my lab is out what were the temporary buildings that were first put up when they were building the original buildings of the Salk Institute. So I had a separate lab sort of within his lab. And as a senior staff scientist, I could apply for my own grant funding and everything. I mean, we worked together, but I had my own independent group, and I had my own postdocs and things within his group, and then interacted with the postdocs in his lab as well.
And you eventually became a research professor.
Yeah. So Dave died in 2020, in August of 2020. So he hadn't really been feeling very well, but because of the pandemic, he was really reluctant to go see a doctor. And by the time he did, it turned out he had B-cell lymphoma, which by that point was untreatable. So he died pretty quickly, actually. I mean, by the time he was diagnosed until he died, it was very quick. The question at that time was, what's going to happen to the lab? And I really wanted to keep the science going for Dave, because he had been the impetus, but behind a significant portion of what was going on. I realized as head of a lab, if I'm not a faculty member, I'm really not part of conversations that are really important to running a lab here. So I asked if I could apply to be a research professor, and they said yes.
If you're enjoying this episode of Beyond Lab Walls, be sure to check out our other channels at salk.edu. There you can join our new exclusive media channel, Salk Streaming, where you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine Inside Salk and join our monthly newsletter to stay up to date on the world within these walls.
So now you're a research professor, you have postdocs, you have science, and you're still looking at a lot of natural compounds. Can you tell me what a flavonoid is?
So we all have certain metabolic processes where we make things like sugar, glucose, and make energy, and we make the building blocks of RNA and DNA, and we make proteins, which is all called primary metabolism. But plants have a whole another set of metabolic pathways called secondary metabolism. And they make thousands of molecules that are not essential for basic survival, but they play all sorts of roles in plants, like protection against various insults, can be attracting pollinators, the colors in flowers. And so among these products of secondary metabolism are a group of compounds called flavonoids, which are what are called polyphenols. They're a group of 3 ring structures. The plants make 6,000 or 8,000 different ones of these.
Polyphenols are small chemical compounds found in nature. Each are made up of carbon rings and other chemical elements like oxygen and hydrogen. Oh, interesting. So we're actually eating flavonoids all the time?
So you can actually, depending on the flavonoid, get a fair amount in your diet. People were interested in them as potentially as antioxidants, but it turns out they do a lot more than that. And the antioxidant activity is probably not particularly relevant in humans because you just don't eat that much of it. In the plants, it may be relevant because they're concentrated in the plant cells.
And what drew you to study flavonoids specifically?
It turns out that they have one of the things that intrigued me about them originally and that still interests me is that they seem to have multiple activities. So they are able to interact with several different targets that are relevant to a number of age-related diseases. Historically, mainly plants were the source of all medicines. For thousands of years, different groups identified plants that could provide beneficial effects, and they knew which ones were helpful for fever or which ones were helpful if you had chest problems or which ones would be helpful in childbirth. A number of our drugs that we still use were based on these, like aspirin is based on willows. Some of the heart medicines, digitalis comes from digoxin, which is a plant product. So there's this long history of use, but chemists had turned medicinal chemists away from this. They made lots of synthetic chemicals. And one of the problems with the flavonoids, they would stick in these dishes non-specifically and give false positive results. So a lot of medicinal chemists those are horrible to use because they give all these false positive results. But it doesn't mean there's not anything wrong with them. It turns out now there's been a return to interest in natural products because chemists have now realized that there are a lot of what are called the scaffolds of different chemical structures. So the sort of the basic architecture. Plants have actually conceived many more than chemists have been able to conceive. So even if the final drug candidate isn't identical to the natural product provides a really good starting point. And so that's one of the avenues we've taken. We've both been interested in the natural products themselves, but also made what we call derivatives, which are based on them, but now are what are called new chemical entities.
So one fruit you've been looking at in particular is strawberries, which produce the flavonoid fisetin.
I studied that molecule for a long time. It's not that abundant in strawberries, but that was kind of a selling point at one point. But you would have to eat quite a lot to get the kind of doses that seem to have effects in humans. But it was rediscovered in the context of aging as what's called a senolytic. So as organisms age, one of the consequences can be that cells develop senescent cells. So these are cells that are not dead, but they've stopped dividing and they've changed what's called their phenotype. So they're no longer doing their normal day job in whatever tissue they're in. Instead, they start secreting a number of factors that can be damaging to other cells. And so some people think that senescent cells are the drivers of aging. I think others think they are a contributor, but it's more complicated than that. And I would come down on that side. And there have been a number of studies now testing it. So for a clinical trial where it's a natural product and you don't have to go through all of the toxicology studies, I'm not sure it's actually worked very well. I do think it has potential.
In addition to flavonoids like fisetin, Pam also looks at cannabinol. Cannabinol is a molecule derived from the cannabis plant and is commonly referred to as CBN. Like CBD, cannabinol is non-psychoactive, and studies have begun emerging that demonstrate cannabinol may be a good tool for managing anxiety and pain. And how's your research on cannabinol going?
We did a study in aging mice, and not so much for the effects, again, more in the context of loss of cognitive function. And there seem to be some beneficial effects in giving the mice CBN, cannabinol. We have a collaborator, Kim Finley, who works at San Diego State, who's an expert on Drosophila on flies. So we've been doing some studies with the CBN and some of these derivatives in flies. You can do assays a lot quicker with flies in terms of effects on aging or effects on other types of neurological insults that she has developed. I was contacted by a company up in Oregon called FloraWorks that is interested in CBN and related natural products. And so I've been testing some of the ones that they have in our cell-based assays. And they're interested in potentially identifying something that they could eventually test in humans. So I'm not sure what the regulatory environment would be in terms of what kinds of approvals would be needed to take CBN into to humans. But hopefully, since it's also a natural product and it's not psychoactive, it wouldn't be too difficult or something related.
What does it take to bring something from your lab to clinical trials?
One of our approaches has been to take natural products and to work with chemists to modify them to improve their efficacy and their ability to get into the brain. So Dave's work was focused on this curcumin derivative he called J147, and he licensed that to a small company, and they took it into phase 1 trials.
Curcumin is a polyphenol derived from turmeric.
Originally, it was for Alzheimer's disease, but it looks as if now they're switching focus to stroke for various reasons. To take something into humans, you first have to get what's called IND approval, investigational new drug approval, which involves a whole bunch of both cell-based assays to look at various aspects of toxicology and inhibition or effects on various enzymes that are involved in drug metabolism, as well as animal toxicology studies that have to be done in 2 different species. And if those are clean, then you can apply to the FDA to get this IND approval, and that allows you to go into at least phase 1 clinical trials, which is where in humans you test safety and tolerability.
Have there been any other derivatives since curcumin?
We developed a number of fisetin derivatives and ended up focusing on one of them, which we called CMS 121. I then ended up partnering with another scientist, Bill Raschke, who had a very small company here in San Diego, and we managed to fund research on first the fisetin derivatives in general, and we are just finishing up a phase 1 clinical trial of CMS 121, and we're trying to get funds to go into a phase 2 trial where we would test efficacy of the compound in Alzheimer's. But it's a long road. It's expensive, but it's been very much a learning experience. Which is good.
So then the real question is, since the drugs are going to take a while, how many turmeric lattes and strawberries do I need to consume to become immortal?
Well, as I said, for instance, fisetin is available as a supplement. You can buy it. So I would suggest potentially taking that. With respect to both aging and Alzheimer's disease, there are genetics involved, but there's also physiological factors, lifestyle factors, environmental factors, all of those that appear to play a role in disease development. And those are factors that you can pay attention to and in many cases may adjust. In terms of physiological factors related to Alzheimer's disease, but related to aging as well, obesity is a risk factor, type 2 diabetes. In terms of lifestyle, getting exercise, eating a healthier diet, both of those play into both aging and Alzheimer's disease, as well as other age-related diseases. Social interactions are important. And I read a study a year or so ago that people who read rather than just watching TV or watching movies or something, they had better cognitive performance as they aged.
Wow, really?
Just because when you read, particularly, and this was particularly fiction, so when you read novels, you have to keep all these characters in your head, and you have to remember what they did 5 chapters ago, and all of that would really makes you have to have much more cognitive engagement than you realize. Another study I read recently that as people develop sort of cognitive problems, they tend to move from fiction to nonfiction because nonfiction is much more straightforward and you don't have to keep track of all this stuff that happened 5 or 10 chapters ago. So all of these can contribute not only to maintaining cognitive function, but also healthier aging as well.
That's so interesting. Do you have other advice for promoting health and healthy aging?
One of the things coming out of a lot of research now is that some of these changes, particularly, well, in the context of Alzheimer's, but this would be relevant to aging as well, again, is that even though we think of Alzheimer's as an old person's disease, a lot of these physiological factors, say as diabetes, obesity, atrial fibrillation, the biggest impact they have is actually when you're, say, have type 2 diabetes in midlife or obese in midlife, not when you're older. So it's actually something you need to pay attention to. And that's something that's not getting out there. And I think it's really important that going forwards that people realize that maybe the diseases manifest themselves later, but your body is setting the stage for that much sooner.
Speaking of the future and going forwards, how do you like running your own lab and helping out fledgling scientists?
I enjoy working with them and seeing them develop from often when they come. I mean, even though they have gotten a PhD, sometimes they're still pretty unformed, I guess, and helping them to learn to write. And writing is critical to success in science. And if you can't convey your ideas well, no matter how good the ideas are, you're going to struggle.
From Connecticut to Canada to San Diego, flavonoids, benchwork, mentoring, you've accomplished so much. Do you have any free time, or what do you do if you find yourself with a little bit of it?
I do a lot of gardening. I like hiking, so I do a fair amount of hiking. Yeah, we have a really wide range of native plants here. San Diego County is actually is the most diverse county in the U.S. botanically. I think probably because we stretch from the ocean to the desert. So there's a lot of interesting native plants. And that's also, particularly this year, has been really fun on hikes because so many things are blooming this year. And there's been waves of different types of wildflower or different blooming plants.
Wow, I had no idea. That sounds very beautiful. I'll have to check it out. Thank you so much for talking to me. It was great getting to know you, your science, your life. I'm glad we got to have you on the podcast.
Well, thanks.
Beyond Lab Walls is a production of the Salk Office of Communications. To hear the latest science stories coming out of Salk, subscribe to our podcast and visit salk.edu to join our new exclusive media channel, Salk Streaming. There, you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine, Inside Salk, and join our monthly newsletter to stay up to date on the world within these walls.